Good morning, everyone. Let's come to the regulation of eukaryotic gene expression today. It's an important part of molecular biology course. So there are multi-level eukaryotic gene regulation. What you'll finding in eukaryotes when you won't find the similar kind of different multi-level organization in the prokaryotes. So that you'll be learning about the different types of regulation in eukaryote, gene structures in eukaryotes, features of eukaryotic genome, and multi-level gene regulation. You see. When the DNA is transcribed into RNA, then it's a capping, and as we have been discussing, the protein is formed and there is a ubiquitination of the protein and degradation of protein. So what kind of signals what we are finding? That is much more important in the regulation of the gene, right? So you see there are different opportunities to control the gene expression in the eukaryotic cell. Okay. Now, as you know, you have the enhancer region, you have proximal elements, and you have promoter regions. All these are important in the regulation of the RNA, especially most of the you would find in generally in prokaryotes. You will generally have promoter, but when you take a eukaryotic region, you have different types of region, like cis elements and trans elements, or for example, if you have enhancer region, some distal control elements, and so on. So gene expression generally you can categorize into very simple way what's called spatial and temporal. So spatial means which regions of the genome is expressed. Okay, not every gene product is important in every cell type. And temporal means where in which time the gene has to be expressed. Okay, whether in terms of environmental stimuli or hormones, or especially seen in the development of formation of tissues and organs, what you will find in Hog genes and Salk genes. So what you will find in the spatial and temporal expression, for example, spatial is you will find in tubulin plants and microtubule found in different places. On the same way, what you will find in the temporal region is globin genes, okay, then tetramer, then add to the heme group, and sometimes in the embryo, or hog gene, and hog, all the homeobog genes which is involved in the developmental phases. So gene regulation at the DNA level. Let's come to the DNA level. Okay, so DNA, if you have any change in the topology of the structure, okay, then it becomes a hypersensitive site for DNA as one. Okay, and they causes DNA methylation. Now once your DNA methylation has been found, you find all the guanine which is added to the cytosine group, and that causes a CG nucleotides in such a way, so it will form CPG islands, and which is not actually transcribing for the protein. Okay, so this all kind of methylated islands what you'll find, which is repressed transcription. So for example, if you see housekeeping gene, a gene is involved in the basic function of all the protein, and there are certain kind of genes which are luxury genes, which for specialized functions, and they are usually in large amount in particular cell. Now another type of modifications what you find in DNA is what's called histone modification, where you have methylation, then you have acetylation, certain kind of acetylations or acetyl group which is added to the particular histone marks are there, where your acetyl groups are added. Then again it makes a protein which is important for transcriptional modification or silencing. So changes of nucleosome, you have there's something called HMG group or high mobility group, okay, non-histone chromatin proteins and so on. You will find high content of charged amino acid, aspartate and glutamate, what you will find. So you will find such kind of nucleosome changes. Then other regulation at DNA level you will find in gene deletion, duplication, and DNA arrangement, which you would always find in at different levels. So transcriptional regulation, what you will find is there are certain kind of elements which is stays or which is harbored at the upstream of the coding region. You have cis elements. So promoter elements which is -35 region, or in the prokaryote, or CAD box or GC box, what you are finding at the proximal elements at the promoter. Then you are finding some enhancer regions or silencer region which is in the DNA. These are all of the elements are important in transcribing the protein. For example, if you find like some protein which is coding for some DNA is coding for some protein. So it's not only the promoter which is important here, you see. So you have a promoter, but also there are some other regions like enhancer regions, silencer regions, okay, different types of or there are some co-activators or different types of elements which is there in the DNA, which is actually deciding the fate of that gene whether to express or not. Then other types of blockers or other type of elements, what's called insulator. Okay, you have a enhancer and a promoter which helps in transcription. Then another set of elements where it's there, where you find insulator which blocks the enhancer action in such a way so the DNA is not produced. So these are all the regulation how a DNA has to be regulated, right? Then you have a trans-acting factor which is not you're not finding at the proximal or distal region of the DNA. So the trans-activating factors are those factors which is controlling the gene expression which is far away from that control center. So maybe expressed in different types of specific tissue or in specific time of the development or in protein modification, or can be activated ligand binding. So you have a eukaryotic RNA polymerases, okay, and transcription factors or domain acting factors or different types of domains or transcription activating domains you will find. So again, you have different types of transcription factor motifs like zinc finger motifs or helix turn, helix leucine zippers and so on. So all these kind of helix, all these which binds to the DNA, which helps in regulating the your DNA. You have leucine zippers and so on. Okay, then as you have been learning about post-transcriptional regulation, you have a exon shuffling which happens, or alternative gene splicing which often happens in the DNA. For example, if you find the same gene, polyadenylation or splicing of different types of, you see there is 1, 2, 3. Then again you have 1, 2, 3, and you have your alternative polyadenylation which is taking place. Okay, different types of polypeptides which is formed. So these are different levels or different planes at which DNA has been transcribed, or transcription which has been happening, or all kind of regulation which is happening at a DNA, so to produce some sort of protein. Okay, then you have RNA interference. See, for example, you have this petunia. So the left is a wild type. Then you have right petunia, contains a transgene that induced to suppress the post transgenes and so on. Then you have unpigmented white flowers, so on. So all these regulations are very much complex in nature, and people are still finding how it is regulating. Okay, I hope you understood. So you have a regulation for protein stability and so on. Okay, I hope you understand. So why we have to learn about the regulations, gene regulation? For example, it's important in the cancers, or to understand what's the oncogene and proto-oncogene, and how we can develop such kind of drugs to come up with the regulation, and what we can express or we could switch off. Okay, so these are the importance of regulation. Okay, thank you so much for your attention.